Hi there. According to the recent African origin model, after Homo sapiens left Africa, humans gradually spread across the world. And along the way, different human populations were geographically separated for long periods of time. For example, the Homo sapiens who remained in Africa and the humans who eventually reached the Americas were separated for a very long time. So, why could they still interbreed? Why didn't humans develop reproductive isolation? Today, let's talk about what reproductive isolation really means. Reproductive isolation is one of the key evolutionary mechanisms behind speciation. It prevents members of different species from producing offspring, or it makes sure that even if offspring are produced, they cannot reproduce successfully. In this way, reproductive isolation helps maintain the boundaries between species. But here is the important point. Reproductive isolation does not always mean two species can never mate or produce offspring. It means that if they do produce hybrid offspring, those offspring usually cannot reproduce normally. For example, imagine a cat and a mouse. One is a feline and the other is a rodent. Even if somehow they actually succeeded, the baby might look something like this. But that offspring would definitely be sterile, because they are not the same species. That is reproductive isolation. Here is another example. Dogs can look extremely different from one another. A corgi and a husky, for instance, are very different in body size. But they can still produce offspring, such as a corgi-husky mix. The real question is, how exactly do they manage to do that? Even though they look very different, a corgi-husky mix can still reproduce. That means corgis and huskies still belong to the same species. But lions and tigers are not the same species. So why can they produce ligers? And why can there even be lion ligers or tiger ligers? To understand that, we need to look at the different mechanisms of reproductive isolation. In simple terms, reproductive isolation can be divided into two major types: prezygotic isolation and postzygotic isolation. A zygote is a fertilized egg. So, prezygotic isolation means that before we even talk about whether reproduction can succeed, the two organisms may not even get the chance to try. Prezygotic isolation mainly includes habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. Let's start with habitat isolation. Habitat isolation means that under certain conditions, one population of a species moves into a different environment and continues reproducing there. It no longer lives in the same habitat as other populations of the same species. They may not be physically very far apart, but because their lifestyles are different, they simply do not encounter each other. Of course, this can also be caused by geographic barriers. Rivers, mountains, and rift valleys can all separate populations of the same species and cause them to evolve in different directions over time. Next, we have temporal isolation. Temporal isolation usually has to do with differences in mating seasons or breeding seasons. In plants, it can mean differences in flowering time or pollination time. For example, two species of frogs may live in the same area. But one species breeds from January to March, while the other breeds from March to May. That makes things a little awkward. One is basically saying, "I haven't even started yet," while the other is already finished. Then there is behavioral isolation. Behavioral isolation mainly refers to differences in courtship behavior. This is especially common in birds and insects. Some species rely on mating dances, call frequencies, or vocal tones to tell whether the other individual is the same species or not. That way, they do not choose the wrong partner. To use a human example, imagine everyone else going out to meet people, while you stay home every day waiting for someone to magically show up. In a way, that could almost count as behavioral isolation. Next is mechanical isolation. Even among dragonflies, different species can have very different reproductive structures. If they choose the wrong species, either the parts do not fit, or they fit but cannot function properly. Another type is gametic isolation. This one is simple. The sperm and egg from different species simply cannot fuse to form a fertilized egg. If all the examples above are cases where the organisms never really get a chance to succeed, then postzygotic isolation is when fertilization does happen. But the hybrid offspring suffers from hybrid inviability, hybrid sterility, or hybrid breakdown. Let's use ligers as an example. Lions and tigers both belong to the cat family, Felidae, and the genus Panthera. They are closely related species. In the wild, they usually do not encounter each other, and their lifestyles are different. That is why most lion-tiger hybrids are produced in captivity. When closely related species hybridize, their chromosome numbers and chromosome structures may be similar enough that there is a chance of producing hybrid offspring. Typical examples include mules, which are produced by donkeys and horses, and growler bears, also known as Pizzly bears, which are produced by grizzly bears and polar bears. Growler bears can be fertile, which suggests that grizzly bears and polar bears have not been separated for very long in evolutionary terms. A liger is the offspring of a male lion and a female tiger. In the opposite case, when the father is a tiger and the mother is a lion, the hybrid is called a tigon. The success rate for producing ligers is very low, around 1 to 2%. Because they lack certain growth-inhibiting genetic controls, ligers can grow to an unusually large size. However, many ligers that are born successfully may die early because of genetic problems. Even when they survive, their lifespan is often limited. Lions and tigers both have 19 pairs of chromosomes, or 38 chromosomes in total. So their first-generation hybrid offspring also have 19 pairs or 38 chromosomes. In theory, if those hybrids mate again with a lion or a tiger, the offspring would still have the same chromosome number. From a biological point of view, it may look like they should be able to keep reproducing. But in reality, male ligers are usually sterile, while female ligers may have some fertility. That is why, very occasionally, lion ligers or tiger ligers can be produced. This brings us to a principle called Haldane's rule. Haldane's rule states that when closely related species or subspecies hybridize, if one sex in the hybrid offspring is sterile or absent, it is usually the heterogametic sex. In species with an XY sex determination system, that usually means the male. In species with a ZW sex determination system, that usually means the female. This is because when hybrid offspring produce gametes, the heterogametic sex is more likely to have problems caused by differences in sex chromosomes. These problems can lead to errors in chromosome segregation and prevent genes from recombining normally. Although there have been extremely rare cases where mules or ligers produced offspring, these are exceptional cases. In general, their descendants still face the problems of inviability, sterility, or hybrid breakdown. For a species, diversity matters, but stability also matters. So a single species may begin to evolve separately at some point for many different reasons. And once enough genetic differences accumulate, those populations may eventually become two separate species. So how long does that process take? Humans and chimpanzees split from a common ancestor roughly 6 million years ago. Both belong to the order Primates and the family Hominidae, but chimpanzees belong to the genus Pan, while humans belong to the genus Homo. In other words, humans and chimpanzees are two closely related species. In 1925, long before modern genetics was well understood, a Soviet scientist carried out highly unethical experiments involving humans and chimpanzees. I will not go into the details here. The result, in any case, was failure. What about lions and tigers? Within the genus Panthera, tigers are more closely related to snow leopards, with their lineages splitting around 3.9 million years ago. Lions are more closely related to leopards, with their lineages splitting roughly between 3.1 million and 1.9 million years ago. As for exactly when lions and tigers split from each other, I do not know. Now let's come back to humans. Homo sapiens appeared only about 200,000 years ago. In terms of speciation, that is an extremely short amount of time. Even though human populations experience temporary geographic isolation, and even though people from different regions today may have different skin colors and physical traits, we are still the same human species. And after the rise of civilization and transportation, human populations across different continents began interacting more and more frequently. Biologically, there is no reproductive barrier between them. So even if humans were given another 6 million years, it would still be very unlikely for us to split into another human species, as long as global populations continue to interact. So let's summarize.
Reproductive isolation does not simply mean that two species cannot mate.
The key question is whether their hybrid offspring can reproduce normally. It is a concept used to distinguish species, not human races. Humans today have not developed reproductive isolation because the time since our populations separated has been too short. And as long as humans continue to interact and interbreed, true reproductive isolation is extremely unlikely to happen in the future.
That is it for today. Stay curious with me.
I'll see you next time. Bye.